Yeah.
Alright, so hi, I'm Ryan Sarkeesian,
I'm Sid Parker.
and this is our presentation on animal testing.
What is animal testing? Some consider animal testing to be the act of harming our furry friends. Others see it as a means of furthering scientific endeavors.
But what is the purpose of animal testing? Many believe that is needed to, many believe it is needed to gather data on how a drug affects like a living system or a human, while others believe it is inhumane and there are other methods that are sufficient and should replace it.
Generally speaking, though, the English College Dictionary simply says it's the use of animals in experiments, which encompasses a wide variety of things that fall under animal testing, from testing medical drugs to the products we use today to agriculture. So just a little quick history. Rachel Pajar mentions how some of the pioneers of animal testing were the Greeks, the Romans, and the Arabians, who used live animal testing to further their understanding of anatomy, physiology, pathology, and pharmacology.
Alright, so some of the experiments that were done that hit different aspects were a Greek philosopher named Galen. He used vivisections and dissections to help us have a further understanding of the anatomy of humans. Avon Zor used animal testing to practice procedures. He did it on animals before he did on humans, so he'd be able to perfect it. Edward Jenner used cow puss to find the cure for smallpox, and the Soviet Union used animals to determine how space would affect a living thing as they go up there. They sent the first dog in orbit, and his name was Lakia. And they also used animal testing to find the effect, to find different functions of the brain. For example, Pavlov used dogs in his classical conditioning experiments, while Skinner used rats in his awkward conditioning experiments.
A quick warning about the next slide here. There's also a lot of animal testing that went a little awry. Some of the crueler and unethical experiments that happened were Vladimir Demikov's two-headed dog, where he performed an entire head transplant by connecting the second dog's head to a host body, connecting their circulatory systems together. Robert White is another famous one. He also did head transplants, but that was between primates. And then Sergei Brakonnikov studied how to keep organs functional outside of the body. And his most famous experiment was a severed dog head that he kept alive for several hours. And these are just a few of the more famous ones. There was also Carl Weinhold's kitten experiments, Harlow's monkey experiments, and Robert Cornish's Lazarus dogs. And none of these animals lasted very long.
Alright, so here are the current regulations and organizations that are involved in animal testing. So first off, the Animal Welfare Act of 1966, it regulates the treatment and care of animals in research, transportation, exhibition, and by the dealers. It sets up the Institution of Animal Care and Use Committee at all these federal research facilities. They are to review all animal protocols and to determine if they are ethical or not. In major research institutions, the Association for Assessment and Accreditation of Laboratory Animal Care International reviews animal protocols in terms of if they are humane or not, but for them, they have to have permission to review the protocols, which makes it kind of sketchy. And then Public Health Service Policy. This policy refers to all invertebrates that are used in research that is federally funded by the Public Health Service Committee. And then the USDA and Animal Planting Inspection Center keep track of all the numbers of how many animals in research, and this is according to Proton.org. So So, so animal testing is needed in the medical community for many reasons. First off, animal testing produces comparable results that are to humans. It gives insight into human physiology and disease, and last, alternative methods are not as effective as animal testing.
12 million to 27 million is the amount of animal deaths via animal testing per year in America alone. That is a significant amount of lives to be lost when the results of animal testing are not nearly as accurate as they should be.
So, animals used are very similar to humans, allowing for similar results. Many of the animals used are susceptible to diseases like cancer and diabetes, as humans are. They are also biologically similar. Mice, for example, share 98% of DNA with humans and have a short lifespan, which makes them easily researched on from birth to death, and from generation to generation. Rachel Hajar mentions that 9 out of 10 experimental drugs fail in clinical studies because we cannot accurately predict how they will behave in people based on laboratory and animal studies. Ryan previously said that mice share 98% of our DNA, but to quote Wesley J. Smith, we share many of the same genes with lettuce, but that does not make us part salad. Also, animal testing plays a major role in getting insight on human physiology and diseases. As time has gone on, most breakthroughs in diseases and also how the human body works has been attributed to animal testing. For example, there's a study at the University of Szeged where they use animal testing and different animal models to determine the different components of diabetes. From this study, they were find out the different complications that come with animal testing. Also, a cure that was found was the polio cure. With this, monkeys were given disease and then released into a community of uninfected monkeys. Researchers then studied them and looked at how the disease spread throughout the community, and this gave them a model what they then used to find the polio vaccine and the cure. And finally, in physiology, Frederick Banks and Charles Best used animal testing to discover the role of the pancreas. On this, they removed the pancreas of one dog and found that the glycocytic levels of the dog increased. And then they took 2 other dogs, removed the pancreas of the other, and stopped the function and closed off the pancreas of the other one. They then used the pancreas of the removed dog and made into a solution, which they then injected into the pancreas of the dog with non-functioning pancreas. This decreased the glycocytic levels and returned the dog back to normalcy without any diabetic symptoms. So, also in vitro methods would not be able to do this, as they are not as effective as animal testing. This because a living system is very complex and hard to replicate. This is according to the California Biological Research Association. In vitro methods are unable to show how like a drug would be introduced to a cell. When a cell is in a petri dish, as in vitro is, the cell is easy access to the cell. But when the cell is in a living body, there's many mechanisms and barriers that the drug has to go through to get to the cell, which is hard to replicate in vitro. There are also different barriers in the living systems that in vitro cannot replicate as well. This includes the blood-brain barrier, which regulates what toxins go up to the brain. And finally, how the systems affect the drug. As the drug goes through the system of the body, it picks up chemical species, which affects how the drug works, which leads to unreliable results as it leads to human trials.
Alright, alternative methods. Animal testing generally goes as follows. There's a preliminary research done, followed by evaluation by dosing animals with high concentrations of the drug or product for about 30 to 90 days, cycling through the stage with different animals. This stage generally takes around 4 years. However, alternate methods are both cost-effective and can have quicker results. In fact, there's already a couple of in vitro methods being used today. Alltox.org lists a couple of them. There's the Q-toxic class method, which was approved in 2007. The fixed dose procedure, also approved in 2007. The cell transformation assay, or CTAs, there's a couple of them that have been approved in 2008. There's a sequential testing strategy for eye irritation and corrosion that was passed in 2014. And there's also human cell line activation test, which was passed in 2015. And again, these alternate methods were the in vitro method, which we were talking a little bit about earlier, which was using a smaller scale to conduct research, basically using cells or tissues or organs. There is also the Silico test, which is using computer programs to test out these certain drugs. Currently, while it takes 3 hours of wait time, and 1,000 processors, which is still faster than animal testing, Pacini explains that we can map out an entire human heart and study the effects of internal and external stimuli.
Alright, and regulations currently in place make sure that animal testing is humane. As I said before, the Animal Welfare Act regulates the treatment and care of animals in transportation, research, exhibition, and by the dealers of the animals. It also sets up the IACUC in the research facilities to review their protocol, and the major research institutions have the ability to have the accreditation of Laboratory Animal Care International, the AALAC, to review their protocols to determine if they're humane. And the Public Health Service policy makes sure that all federally funded research by the Public Health Committee is regulated and humane.
Alright, so I'm going to talk a little bit about the rabbit pyrogen test as an example of an in vitro method that is safer and better. So pyrogen is a bacteria that can cause fever if entered into the bloodstream. Generally speaking, rabbits are used to test for this pyrogen because their body temperature rises significantly and it's quite noticeable. However, as Danishad and al. mentioned, 100,000 rabbits die yearly in Europe alone from this test. The in vitro method that counteracts this, or should be used in place of, is the LAL, or these are blood cells extracted from horseshoe crabs, and while horseshoe crabs are not technically protected under the acts, they're still thrown back into the ocean after their blood is collected. So there is a lot of promise with in vitro methods. There's already quick and successful methods out there that can do the same things as animal testing but cheaper. And so again, I've thrown a lot about how in vitro methods are cheaper. Here's just a couple of price examples. Generally speaking, a lot of them are thousands of dollars cheaper than animal testing. Meggs and al. States that currently Europe spends around 1,300 million pounds, or the equivalent of 1,607.52 million US dollars yearly on animal testing alone. And the US drug industry, as Cruelty Free International also states, says the US spends around $50 billion in research, but has about the same approval rate in new drugs as it was 50 years ago.
So animal testing should not be banned because of the reasons that animal testing produces comparable results to humans, and that the alternative to animal testing are not as effective in producing those results. It gives crucial insight into human diseases and physiology, and that other methods and that the current regulations in place are effective in regulating that is humane and not stepping over any boundaries.
So I argue that animal testing should be banned. These tests do not fully represent the human body, and they're not as accurate as, again, as they should be at this point in time. And animal testing would be surpassed by better and alternative methods in the future. And lastly, animal testing is inhumane. And then here's a couple of our references.